So the topic of my presentation under nanotechnology is compare and contrast light microscopy with electron microscopy. So starting with the introduction part, first we have to know what is a microscope. So a microscope is a scientific instrument which is used to magnify objects too small to be seen with the naked eye. It actually provides us with enhanced resolution, contrast and magnification. And what is microscopy? Microscopy is a scientific technique and technical field of using microscopes, like how we use the microscope. A microscope is consists of structural components like the base, arm, and head, and optical components including the eyepiece, objective lenses, adjustment knob, etc. So there are two broad categories of microscopy we are concerned with. That is the light microscopy and the electron microscopy. So in the later slides, we will be discussing about light microscopy and electron microscopy. Here, if we look at the differences between light microscopy with electron microscopy, light microscopy is also known as optical microscopy, whereas electron microscopy is also known as beam microscopy. Light microscopy is known as optical microscopy because it uses light, which is approximately of 400 to 700 nanometers. The wavelength of light is 400 to 700 nanometers. It illuminates the object under view. And in electron microscope, it uses a beam of electron. The approximate wavelength of the electrons is 1 nanometer to make objects larger for a detailed view. In light microscopy, the image is formed by the absorption of light waves. In electron microscopy, the image is formed by the scattering or transmission of electrons. Light microscopes are smaller and lighter, whereas electron microscopes are heavier and larger in size. In light microscope, lenses are made of glass, but in electron microscopy, lenses are made of electromagnets. Basically, the light microscopes are not used under a vacuum, but in case of the electron microscopy, it is been operated under a high vacuum. There are some types of light microscopy. Now, coming to the types of light microscopy, first we have bright field microscopy. A bright field microscope is a compound light microscope and it produces a dark image of the specimen against a bright background. So it uses transmitted white light to produce a dark image of the specimen against a bright background. It works by sending light through a condenser and then the specimen. The specimen's denser areas absorb and scatter the light, causing them to appear darker against the uniformly bright background. This simple and affordable light microscopy, which is the bright field microscopy, requires straightforward sample preparation and it is widely used in the fields like medical diagnostics, food science, and soil science because it is very easy to use and easy to handle. After bright field microscopy, we have another type of light microscopy, that is the dark field microscopy. So in dark field microscopy, the microscopes creates a dark background to allow viewing of small unstained objects. In the bright field microscopy, we have used stained objects, but in dark field microscopy, we are using unstained objects such as the motile bacteria that would be difficult to view under a bright field. The central portion of the light in dark field microscopy is blocked so that only oblique light strikes the specimen and scattering light rays that then enter the objective to form the image that we will be able to see through the microscope. The dark field microscopy is a method from the 19th century. In this type of microscopy, the specimen is bright while the background is dark. The dark field microscopy is high contrast, good for unstained and live model specimens. Below are some pictures under the dark field microscopy. The next type of light microscopy is the phase contrast microscope. The contrast microscopes make a transparent specimen like unstained living cells visible by converting the invisible phase shifts in light into visible brightness changes. It actually uses a special annular diaphragm and phase plate to achieve this. It allows researcher to observe cellular structures and activity in detail without need for staining. The mechanism of Phase contrast microscopy is such that in phase contrast microscopy, the central portion of the light source is blocked, which creates a ring of light from the condenser that illuminates the specimen. The light waves reflected by the specimen are slowed by a phase retardation plate, and it increases the difference in wavelength between refracted and unrefracted rays. When the refracted and unrefracted waves are focused, they produce an interference due to the difference in wavelengths. The difference in wavelength produced, this is actually seen as differences in brightness in the specimen. This is a picture under a phase contrast microscope. Next, the type of light microscopy we have is the polarization microscopy. So, the polarization microscopy uses polarized light to reveal the optical properties and the three-dimensional structures of the anisotropic materials. Now, the anisotropic materials are those materials which are dependent on the direction. If we discuss about the working, a polarizing microscope works by passing plain polarized light through an anisotropic specimen. The plane polarized lights, when passes through the anisotropic specimen, it splits the light into two rays that travel at different speeds. These two rays then pass through a second polarizer called the analyzer. The analyzer is set at 90-degree angle to the first polarizer. This arrangement This type of arrangement creates a high contrast image by exploiting the birefringes and interference, which reveals the specimen's composition and its three-dimensional structure. Below, I have attached some pictures of the specimens under polarization microscopy. The next type of microscopy we have under light microscopy is the fluorescent microscopy. So the fluorescent microscopy allows detection of ions and molecules within the cells. Fluorescent dyes absorb short wavelengths of light and emit longer wavelengths. The working principle of fluorescent microscopy is such that a high energy light source shines on the sample, passing through an excitation filter that allows only specific and short wavelength light to pass through. The fluorophores in the sample absorb this specific short wavelength light. After, in response, the fluorophores emit light at a longer wavelength, a phenomenon which is known as fluorescence. A dichroic mirror reflects the shorter excitation light and allows the longer wavelengths emitted light to pass through the detector. After that, an emission filter blocks any remaining excitation light, allowing only the emitted fluorescent light to reach the detector and form a high contrast image against a dark background. Now we'll discuss about the electron microscopy. So electron microscopy is a type of microscopy technique that uses a beam of electron instead of visible light to illuminate a specimen. Because electrons have much shorter wavelengths than photons, and electron microscope have much greater resolving power because of using electrons, which allows visualization of extremely small structures. There are mainly two types of electron microscopy, such as transmission electron microscopy and scanning electron microscopy. So, transmission electron microscopy is a type of electron microscopy. In transmission electron microscopy, a beam of electron is transmitted through a very thin specimen to form an image because the electrons interact with the internal structure of the specimen. Talking about the working principle of transmission electron microscopy, a beam of very high energy electron is generated by an electron gun and accelerated through a vacuum. The beam is focused using electromagnetic condenser lenses. In case of transmission electron microscopy, the sample must be extremely thin, typically 100 to 200 nanometers, so that electrons can pass through. As electrons cross the sample, some are transmitted, but some others electrons are scattered or absorbed, dependent on the thickness, density, or structure of the specimen. The transmitted electrons are focused by objective projector lenses to form a magnified image on a fluorescent screen or detector. Here is a picture I have added under the transmission electron microscopy of a specimen. The second type of electron microscopy is the scanning electron microscopy. The scanning electron microscopy produces high resolution images of a sample surface by scanning it with a focused beam of electrons, which reveals its topography and composition. So, basically, scanning electron microscopy uh, gives us the image of a sample surface by scanning the surface. Speaking about the working of SEM, a focused electron beam scans the sample surface in a zigzag pattern. The interaction of the electron beam with the sample atoms produces various signals. Detectors collect these signals, including the secondary electrons, which reveal surface details, and the backscattered electrons, which provide information about the composition. The collected signals are processed and dis- displayed on a screen, forming a detailed image of the sample surface. The scanning electron microscopy produces dark images of the specimen. So here is the conclusion of this presentation, that microscopy has fundamentally transformed our ability to observe and understand the microscopic world. From early light microscope that open our eyes to cells and basic tissue structures to advanced electron and fluorescence microscopy revealing ultrastructure, molecular interactions, and dynamics in living systems, the field has continually expanded what is visible and knowable. So, microscopy is not just a tool, but a dynamic suite of technique that together enables scientists to probe structure and functional scale from whole tissues down to molecular assembles. Its continued development and promises even greater insights into biology, medicine, and material science. So, these are the references of the presentation. Thank you.